Hello, welcome to the Science of Emotions podcast. In this episode, we will be discussing the neuroscience of positive emotions and affect. And this is your host, Ruben Singh, MD. It's really important topic and what may be surprisingly quite urgent. Despite happiness and positive feelings being so central to human well-being, the actual neuroscience of positive emotions is still pretty nascent. We've spent decades intensely focused on the neurobiology of fear, stress, anxiety, all the negative stuff. We know so much about what tears us down, but comparatively little about pathways that actually build us up. And that is the gap we want to bridge today. It's a review paper published by Neuroscience and Biobehavioral Reviews. The neuroscience of positive emotions and affect, implications for cultivating happiness and well being. And this is not just one lab's perspective, it's a grand synthesis pulling together what we know currently about the positive brain states. It draws from psychology, philosophy, neurobiology. The question is, can we actually train our brains for sustained well being? That really is the core question. Can we cultivate this? We'll be looking at all the key themes like the neurochemistry behind pure pleasure and motivation, liking versus wanting, and the dynamic, flexible networks in the brain that handle emotions. It's not as simple as we once thought. And then we'll scale up to the impact of social factors, developmental changes, and even specific practices like meditation or getting into flow states. And maybe the best place to start is with the language itself, because the scientific community based on this review actually recommends moving beyond just focusing on happiness. That is really crucial distinction right at the outset. Review strongly suggests prioritizing well-being over the simple pursuit of happiness as a transcendent human goal. The westernized approach often emphasizes chasing those high arousal positive emotions. You know, that ecstatic burst of joy, the immediate gratification, the big win, the peak experience. But that focus can be fleeting. It can eventually, paradoxically, set us up for disappointment when it fades. Well being is broader, more sustainable. So, if we broaden the scope to well being, that takes us back to such really old philosophical ideas, those dualities that these thinkers have been wrestling for centuries. Let's formally define those two core concepts in research, starting with the classic idea of pleasure, that's hedonia. Hedonia, it translates roughly into subjective well-being. Fundamentally, it's about maximizing the positive experience and minimizing the negative ones. It's that moment to moment feeling of good affect, pleasure, content. Epicurus, seeking that calmness, that inner peace by avoiding distress. He called it ataraxia. Or you could think of someone like Jeremy Bentham, the utilitarian view seeking pleasure like wealth, friendship, or just simple relief from pain. Characterized by pleasant affect and overall life satisfaction. So, hedonia is basically how good you feel right now and how satisfied you are with your life generally. Other theorists have since expanded on the specific elements of subjective well being, including traits such as wisdom, knowledge, courage, justice, temperance, and transcendence. Now, the second concept is eudaimonia, that is psychological well being, flourishing. It defines well being in terms of development of human potential. This Greek concept of eudaimonia focuses not just on feeling good, but on doing good, on optimal living. It's about achieving moral and ethical virtues, self realization through purpose and meaning. And it's much more process oriented. It's about how you live your life, not just emotional outcomes at any given moment. Think of Aristotle's ethics. He defined the true happiness, eudaimonia, through cultivating virtues like courage, justice, prudence, that sort of thing. Or maybe Buddhist middle path, the idea of moderation leading to a kind of balanced flourishing. That's the great example, that Buddhist way that points towards enduring state of being, a way of living, not just chasing a momentary rush. What's really fascinating about the modern science seems to be backing this up. The research shows that they're related, hedonia and eudaimonia, but they are empirically distinct. You can be absolutely be hedonically satisfied, say relaxing on a beach, feeling good, but still lack the eudaimonic sense of purpose or feeling fully realized, or vice versa. Even though the psychological and subjective well beings are conceptually related but empirically distinct streams of psychological functioning, these two concepts have overlapping features. Also, conceptualizing eudaimonia and hedonia as distinct orthogonal psychological construct may be detrimental to advancing the theory and research. As one researcher, Todd Kashdan, asked rhetorically, quote, "Have you ever met a parent who said, 'All I want is my kid to have meaning in life without happiness, joy, and sense of feeling alive?' For example, striving for a difficult but meaningful goal involves some hedonic discomfort, but high eudaimonia. And the field has gotten much better at measuring this by integrating these two aspects. And the research has developed sophisticated composite measures like Compass W scale. That's COMPAS W scale mentioned in this review. It measures composure, own worth, mastery, positivity, achievement, and satisfaction. This is an attempt to capture the whole spectrum, both the momentary feeling good and the purposeful living aspect. And the evidence behind it seems pretty compelling. The high scores aren't just about feeling good subjectively. High composite well being measure by the scale is associated with some really tangible benefits. Things like superior working memory capacity, better attention regulation, good sleep quality, even lower work absenteeism that connects psychological health directly to high functioning life. It demonstrates that flourishing isn't just nice to have, it's actually an adaptive advantage. If you manage your emotions effectively, keep positive things salient in your mind, you just function better across the board. So to really understand how this works, we need to go deeper, right down to the micro level. the chemistry, the anatomy of how brain actually processes these positive feelings. So let's talk reward. So this review article breaks down the concept of reward into three distinct interactive parts. There's liking, which is actual pleasure of the moment, the enjoyment itself. Then there's wanting, that is the motivation, the drive, what they call incentive salience, the urge to get the reward. And finally, there's a reward based learning, how we associate actions with outcomes. And right there, getting these three distinctions is where we hit the biggest misconception in popular science, especially around dopamine. The dopamine, the famous pleasure chemical. Except that it's primarily wanting molecule, not the liking one. Dopamine drives that widespread messy system, the seeking network. It governs the motivation, approach, behavior, exploration. So the dopamine is what gets you out of the bed to chase that promotion or, go get the cup of coffee or seek that social connection. But the actual moment of enjoyment, that's sipping the coffee, getting the hug, that might be handled elsewhere in the brain. So largely, the dopamine is the engine that drives the search. And that's this really critical point that this review article makes. So this is because there is a serious issue in directly linking dopamine solely to the positive affect or pleasure. Much of the evidence linking it to pleasure is inferential. It's based on observing reward seeking behavior, correlational studies. It's not usually a direct measurement of subjective enjoyment. It tells you what an organism seeks very strongly, but not necessarily what it truly likes in the moment of consumption. So, if dopamine handles the powerful wanting, the drive, what handles the pure, unadulterated liking, the actual sensation of pleasure? Now, that seems to fall primarily to the brain's opioid system and their receptors. And these are concentrated in areas called hedonic hotspots. The hedonic hotspots are specific locations, incredibly specific, highly localized, precise anatomical structures. Animal research shows that you can do micro injections and really pinpoint things that has identified these pleasure generating hotspots deep in the brain, notably in nucleus accumbens and also in posterior ventral pallidum. We are talking an astonishing level of precision in rat models. These key hotspots where opioid stimulation really boosts pleasure measures just 1 cubic millimeter in volume. It's incredibly tiny. And when opioids are stimulated, especially in that tiny space using, say, a mu opioid agonist, it can more than double the intensity of liking reaction. Things like the lip smacking response in rat shows to the sweet taste. But if you stimulate dopamine outside this tiny hotspot or even nearby, you increase wanting. The animal still works harder, seeks more of the sweet reward, but enjoyment doesn't increase. actual liking reaction, the pleasure expression itself doesn't intensify. It highlights this dissociation between drive, dopamine, and the pleasure, opioids, in specific hotspots. A tiny cubic millimeter is where your pure biological pleasure gets amplified. That specificity is quite incredible. Now, brain's pleasure circuits are not just about location, they're also highly dependent on context, which explain why that fifth cookie is not as good as the first. Context is the key. Orbital frontal cortex, that's OFC, is the critical here. It seems to track our substantive experience of pleasure. It integrates not just the stimulus itself, but the context, our internal state, the level of satiation. There is a classical human study that illustrates this very well. Participants drank a pleasurable reward like chocolate milk repeatedly while on a scanner. The neuroimaging showed that as they drank it to satiety, as they got full, the pleasure naturally diminished, and activity in their OFC was also subsided. Less pleasure, less OFC activity. But then there's a really neat part of the study. Then they immediately switch to a completely different, unhabituated drink. Say something like unexpected, like tomato juice. Now, then the OFC activity jumped right back up. It showed a renewed pleasure process, even for a stimulus that might not be normally their top choice, simply because it was novel relative to the now boring chocolate milk. So the OFC, that is the orbital frontal cortex, isn't just is a detector that's saying chocolate milk tastes good. It's saying that this tastes good right now relative to my current state of need and recent exposure. It perfectly explains why variety is the spice of life, neurobiologically speaking. Brain is wired for relative pleasure and novelty detection. It shows pleasure is flexible, homeostatically driven. It is not just an immutable property of the thing itself. So, moving beyond these core reward circuits, review also talks about the role of social neurochemicals. Oxytocin. It is often called the love hormone or bonding chemical, known for its role in the socio emotional function like trust, pair bonding, but is also crucial for social reward. How does it tie into dopamine and opioid system that we just discussed? Well, the theorized mechanism suggests a kind of cascade. Oxytocin seems to attach social salience to the stimuli. It tags things like social connection, a loved one's face, or participation in a group activity as important and worthwhile. So it flags the social interaction as something valuable. It's kind of putting a sticky note on it for the rest of the reward system saying, pay attention to this. And then this social tagging is thought to mediate or even directly stimulate dopamine release further downstream in areas like the ventral tegmental area, that is VTA, and nucleus accumbens. So the oxytocin highlights the social targets, then dopamine provides the motivation, wanting to approach and engage. The oxytocin facilitates the social approach and connection, and this engagement then stimulates the dopamine opioid reward system. This translates into those positive social emotions we cherish: happiness, joy, that powerful sense of belonging, the seeking. The dopamine drives gets us oriented towards social reward because oxytocin made the social connection salient in the first place. So, next, the review article briefly summarizes the role of gonadal hormones like testosterone, estrogen, and progesterone in positive emotion. Now, although the explicit role of gonadal hormones is still largely unknown, yet mounting evidence document the beneficial role of hormone replacement therapy to restore mood and well-being during aging processes. Okay, so now let's move up a level from the chemicals to broader brain anatomy. A key finding is that there's no single happiness center. That's one of the strongest conclusion from the meta-analysis in this review. The old idea, the brain-specific centers for each discrete emotion, it's considered to be too simplistic. So there's no single a region in the brain that uniquely represents positivity or negativity. Instead, fMRI data overwhelmingly supports the existence of what's called flexible affective workspace. Again, that is a flexible affective workspace. It involves a network of interconnected regions. We are talking about the medial prefrontal cortex, that is MPFC, anterior cingulate cortex, ACC, anterior insula, that's AI, and amygdala, and ventral striatum, areas we know are involved in emotion and motivation. The flexible part means that they handle everything. The lesion studies and EEG research has demonstrated that positive affect lateralized to the left PFC and negative affect lateralized to right PFC. So the left dorsolateral PFC contributes to processing positively valence stimuli and emotions. What makes it flexible is that these regions are involved in processing all sorts of affective stimuli, both positive and negative. Their specific role and interaction pattern depends on the context and requirement of the moment. Now, this lines up really well with the constructionist theory of emotions. The idea that emotions are dynamically built or constructed by the brain, not simply triggered in a fixed location. It's interesting that amygdala is in that list. We almost always associate it with fear, the fight or flight response. That's the classical association, but it's incomplete. Amygdala's role is much more dynamic and contextual. It's activated just as reliably by intense positive stimuli like winning a prize or seeing a loved one as it is by negative ones. Basolateral amygdala has a role of updating the representation of value. With regard to positive emotion, the research has suggested that amygdala processes valence in a bipolar manner, with amygdala activity increasing in response to negative stimuli and emotion, and decreasing in response to positive stimuli and emotion. Building on this theory, release of endogenous opioids in amygdala was related to decreased amygdala activation during experience of positive emotions in humans. There's a conflicting research result about whether amygdala tracks valence or arousal, and further research is needed to clarify that issue. Now, its job seems to be processing salience. Uh, Another emotionally significant, motivationally relevant that is critical for the organism, whether it's dangerous or rewarding. It flags things as important for further processing. So it's salience, not just threat. This brings us to affective chronometry. That's the timing of emotional responses. It's called affective chronometry. When we talk about processing emotions, we really have to think in milliseconds. An emotion like joy isn't just one solid state, it unfolds rapidly over time. So researchers mapped these with EEG and MEG from by millisecond, the dynamics of emotional or affective processing. Now, what do these millisecond maps tell us? What does it look like for positive emotions? Emotion specific temporal signatures are often influenced by a range of individual differences. Studying the temporal dynamics, that is, how emotions unfold over time, is critical due to the distinct nature or aspects of emotional responses like reactivity, regulation, and repair that develop across time. Affective chronometry of basic emotions occur during specific but not continuous time segments, with the accumulating evidence that shows that positive affective stimuli are processed later relative to negative affective stimuli. And valence and arousal are associated with distinct patterns of spatiotemporal activity. And presumably, these distinct time windows, these chronometric patterns, might allow scientists to eventually distinguish these neural signatures of, say, happiness from excitement, even if these similar brain areas in that flexible workspace are excited. That's the hope from further research on affective chronometry. So we have discussed chemistry driving the motivation and pleasure, flexible networks processing the feelings, and precise timing. And how do these positive feelings actually impact our ability to think, plan, or control our behavior? That brings us to the next segment of cognitive control. Cognitive control framework is absolutely critical for understanding well being. The cognitive control basically refers to our set of executive functions, things like working memory, inhibition, selective attention. These are the tools we need to guide our behavior towards our goals, especially long term goals. Despite the constant stream of emotional distraction or temptations, I like to think of cognitive controls as brain's internal CEO trying to keep us on emotional employees focus on the quarterly goals. The dual mechanism of control, that is DMC framework, breaks down the CEO strategies into two main approaches. First, you have proactive control, that is the anticipatory, strategic, sustained kind of control. It's preparing in advance, maintaining goals, information activity. Think about diligently planning and saving money over years for retirement. Planning ahead, staying focused on the long term. That is the proactive control. And the second strategy is the reactive control. That is more of ad hoc, transient. It's triggered only when needed in response to an immediate challenge or a sudden reward cue popping up. It's that quick burst of focus you deploy when you say you almost drop your phone and manage to snatch it out of the air just in time. It's reacting effectively in the moment. So, so there are two controls, proactive versus reactive. So how does being in positive mood affect these two control systems? Does feeling good make us better or worse at managing ourselves? Well, the evidence suggests that it's a complex trade off, and it often seems highly dependent on the arousal level of the positive mood. Low arousal positive affect like faint contentment, relaxation, serenity tend to reduce our proactive controls. So feeling good makes us a little bit less strategic. When you're deeply content and relaxed, your brain often shifts into a more diffuse open processing mode. This fits with the idea of some positive emotion broaden our attention and cognitive scope. You might stop rigidly sticking to your present plan. So if I'm feeling super relaxed and satisfied on vacation, I might be less likely to stay laser focused on, say, mapping out a complex project plan for work. So your brain perceives no immediate threat, no urgent, compelling goal that needs that tight, sustained focus. You feel secure, so you broaden out. However, positive mood generally seems to increase reactive control. It helps us respond quickly, efficiently, and often more creatively to immediate rewards and transient challenges that pop up. So, contentment might loosen the rein on long term strategic planning, but makes us quicker and more flexible in dealing with what's right in front of us. But then there's an important layer, a motivational nuance. We can't lump all positive emotions together. There's a critical subtlety. A specific type of positive emotion matters. The research highlights that certain positive emotions seem tied to specific control outcomes. For example, emotions like pride or self worth, which often quite feel high energy, achievement oriented. These states are often associated with increased inhibitory control and better focus on long term goals. They seem to narrow attention back towards the task completion and persistence. So, feeling of pride is less about pure pleasure, but maybe more about signaling goal completion, reinforcement, that effort needed to get there. How is it that different from the simple joy? It seems to be about their motivational focus and context. Pride and self worth often come after overcoming difficulty or achieving something significant, requiring sustained effort and inhibiting distraction along the way. In contrast, emotions like basic happiness or joy or fun are often associated with achieving short term goals or simply presence of an immediate, unexpected reward. In these states, the simple joy, fun, sometimes leads to less self control, maybe more impulsivity. It's why when we feel lighthearted and joy, we might be more likely to give into that immediate temptation like eating more chocolate or skipping the gym rather than sticking to a long-term plan like a diet. So let's connect this to the another executive function, that's working memory, ability to hold information in mind. How does that relate to well-being? The connection here is that really strong and consistent in the research. Better working memory capacity is clearly associated with improved emotional regulation and overall life satisfaction. This link is pretty powerful. Why does working memory do What does working memory do for our emotional lives? Working memory is what allows us to consciously hold, manipulate, and update mental representation. So the ability to effectively update our working memory and importantly to keep positive stimuli salient in our mind, actively remembering a past success, looking forward to a pleasant event. That ability is directly related to increased life satisfaction. You're essentially using your working memory as a tool to actively manage and improve your current affective state. And this is where we see a fascinating pattern develop that we'll discuss in part two of this. That is the positivity bias in older adults, and further interventions that are discussed in this review will be discussed in part two of this episode on positive emotions. So this concludes part one. We'll see you next time.